Thank you. Thank you so much for the invitation. And I hope you won't be disappointed. There is not so much on the fly. Maybe soon. We have a lot of experiment going on at the moment. So I hope people online can hear me well. Looks so. All right. So let's get started. So yeah, I wasn't sure. Like I made this little slide just to introduce myself, but you already summarized it very well. But maybe mostly to say that I started to be interested in biology by being on the field, so by being fascinated by the diversity of life. And then I discovered that with genome, you can go in the hidden diversity. So that's why I really like about being able to do biology from the field to the lab with experiment. You can look at the phenotype and then go into the genome and look at the genetic variation. So yeah, this is a photo where we sample flies on its habitat, the seaweed in Brittany with some members of my groups. All right. So yeah. So I guess, yeah, like as I say, I'm interested in biodiversity, and I guess like most of you in evolutionary biology, we try to understand this origin and evolution of biodiversity from the community level, maybe the diversity of ecosystem, the diversity of species, and below that we have this diversity of traits. And the basic of all of that is genetic variation. So we are trying to link the genetics and the genetic variation to ecology. And sorry, I did something wrong here, and then I can't see my slide. So, okay, I'm back. No worries. All right, so what we're trying to do in evolutionary genomics or genetics is trying to link the phenotype to the genetics and ask how ecological factors impact that through both demographic process and selection, and how all of this interaction shape evolution. So then, where come the structural variant I wanted to tell you about? So we all know that genomes vary in sequence, and we have known for a long time, since people can observe chromosomes, that they also vary in structure. So why are we studying structural variant in evolutionary biology and population genomics? To answer this question, we've been trying to write a kind of beginner's guide with these people. This was mostly led with Katharina. So most of the introduction is based on this review. So that's why I'm introducing that and thanking the co-authors on that. And to start this review, we've tried to start from questions arising from the community, arising from early career researchers saying, okay, what about structural variant? What do people wonder? And to answer this, we've tried to base on the current literature, and we look at a lot of studies, about 100 in ecology and evolution, and you can already see, if that works, yes, from the keywords that we use to do the literature search, that there is quite a diversity of keywords that make it a bit complex sometimes to approach the field of structural variants. So some of the questions that appears are, what are structural variants? Because the definition was usually unclear. So we all know single nucleotide variant, single nucleotide polymorphism. Structural variants are more defined by the way they impact the sequence, and they can be defined as the presence, absence, orientation or position of a given sequence. And then they have different names based on the thing they do in the sequence. There is also a notion of size, and people often take a cutoff at 50 base pair, but that's purely arbitrary and coming from what was doable with bioinformatics a few years ago. So I invite you to think about more range of size. And the biggest one that affects all chromosomes are often called chromosomal rearrangement. And I've put here a few examples. For instance, here it's a 2 kb indel that impact the color of those crows and impact the sexual selection. And here you have a megabase scale inversion that is involved in ecological adaptation in this mimulus plant. And here an example of large scale chromosomal rearrangement, so fusion that differ between the two species of butterflies. So here you can already see one of the challenges that appears. When you look at SNP, it's all homogeneous variants. Here we have a diversity. So how can we classify and analyze this diversity of variants? When I think about SV, sometimes I think, okay, that's this pool of different candies, and how should we analyze them? Together, maybe it doesn't make sense, but then how to split it? One of the answers comes from the bioinformatics. Depending on the signal they leave in the sequence data, they are classified according to their type. From the bioinformatics, we could also get the length, which may be relevant for the impact on phenotype. You can expect that something longer may disrupt more genes. But really, from an evolutionary point of view, maybe we want to classify them a little bit differently. Something we are really interested, not sure we have the answer yet, is to split them by mutational origin, by what formed them. In particular, we know that transposable elements, so you know this element that do copy and paste or cut and paste along the genome, are really mutagenic, like they form insertion and deletion, but they can also lead to ectopic recombination that leads to bigger structural variants. And ideally, what we would like to do for variants is split them by properties. But this, we don't have access to the properties, but ideally to know their fitness effect or to know their impact on recombination or even on dosage. So on dosage, for example, you can think about duplication, copy number variants, because the more copy of a gene, the more you could expect to have RNA transcript. It's not so linear. To show you an example, this is a work I've been doing on the whitefish. And here we have two species, the dwarf and the normal, and we've been detecting structural variants in this species. Here you see the number of structural variants as a function of length, and you can note already that you have the same, a lot of SV of the same size, and when you look at the sequence, it corresponds to some transposable elements. So looking at the sequence is a way to say some of them are due to transposable element or not. When I was saying it could be interesting to look at the structural variant together, in these two studies, I found that super interesting because they look at duplication on small structural variant and big rearrangement. And what they found in the deer mice and the seahorse is that very huge inversion have at their breakpoint a lot of duplication or long inverted repeat. And with this, they kind of infer that maybe the inversion was formed by ectopic recombination. So recombination matching this inverted duplication or similar repeats, and that created the breaks, originated the big rearrangement. So I think there is value in considering this diversity of variant altogether. So one of the questions that also we hear, what is the level of genetic diversity attributable to SV? Maybe we don't care about SV, it's just a few here and there. Well, not so much. When you think about the fraction of the genome covered by SNP versus the fraction covered by SV, you see that much higher, not because they are more SV, but just because they are longer. So here you have a value between human and great apes. They would differ by 1.3% only based on SNP, but much more based on SV. Yet you see this range of 3 to 10 times here. This really not working well. Okay, and this diversity could depend on the studies. Do we have the power to detect the structural variant, or does it depend on the species? And a lot comes from the data. Like in human, when they use short reads and long reads, they wouldn't say you and me differ by the same average number of structural variants. I've looked at that in the whitefish dataset that I showed you earlier. And you can see here that depending on the data set I used, either assembly, long read, or short reads, I do not detect the same number of SV. If I look at the overlap, so one color is one data set and several colors is several data sets, there is limited overlap. And the reason for this is that the different data sets do not offer the same power to detect structural variants. So we need to be very careful if we want to compare one species to the other or one population to the other, because depending on the quality of the data or the type of data, we don't get the same answer. A frequent question is also, why studying structural variant when we already have SNPs? Maybe we don't care, right? Particularly in agronomy, people have a good answer to that, is that structural variant can have an important impact on phenotypes. They try to quantify the heritability added by structural variant, and you see that it's only 29% of explained heritability with SNP and more than 41% with structural variant. So this genetic variation matters. And sometimes we could use linkage. SNPs would be linked to SV, but not always, about half of the cases. It could also be important for conservation genomics. In this study on birds, they try to quantify the proportion of derived allele counts and the impact, the proportion of variants that have deleterious, strong deleterious effect or moderate. And you see that structural variants are in general have a higher deleterious effect and they are more rare. And they're really predicted to be more deleterious than SNPs, so could matter for conservation genomics. And what about in population genomics? So again, coming back to my example of the fish, you see here a PCA done with structural variant and SNP, and each dot is a fish, and they group by lake and by species. So here you would say, okay, we don't care about structural variant, right? To just get the structure. I would have the same with 100 SNP. But it's not the case in all species. And if you ask my friend Jan, who did that in the lobster, he found that with SNP, he see no structure, yeah, while with copy number variant, he would see a geographic structure among its lobster population. So why so? It could be because of CNV evolving slightly faster, or also correlating with some micro-environmental differences. So coming back to this idea of speed, we know that some structural variant can contribute to the evolution of genome quite quickly. In particular, transposable elements, they can work as bursts. They can be very important contributors to genome dynamics, like we talked about this morning, and they can appear and spread quite fast. It seems also to be the case for duplication. Like duplication rate would be higher than the nucleotide mutation rate. So maybe it's not so surprising that we see rapid adaptation associated with duplication. Like, imagine when you duplicate a gene, if it happens quite frequently, of course, it gives a good solution. And then one of the questions is also, what are the properties of structural variants that can matter for evolution? So it's worth knowing that they can have strong indirect effects. For example, they can alter the genome organization and functionality. Maybe you heard about topologically associated domain, like the way the chromatin and the genome interact. All of this seems to be reshaped, at least by some rearrangement, like the fusion. We know that some structural variants will affect methylation, an epigenetic mark, and it will also affect meiosis and pairing. And about that, because I will talk a lot about big inversion, I wanted to mention this case of non-recombining blocks. If you look in the literature, this is only a subset of the recent example, but in many, many species, people find something like they do PCA along the genome, and they find big blocks, like here, where the PCA has the same pattern. And usually this reveals non-recombining haploblocks, so big blocks that do not recombine. And it's not really expected in a wild natural population. So this may be due to large inversion. As a reminder, why of that? Because inversion are supposed to be when heterozygote, making loops during meiosis, which will lead to unbalanced gametes. So the resulting effect is that crossover can happen, but recombination is not achieved. And over time, these two arrangements will slowly diverge and accumulate mutation, and you expect to end up with that. However, it's not the only way of making this pattern, and I would argue that chromosomal rearrangement needs to be confirmed by other data. But it's been the case in several species, and it's very interesting to see that this non-recombining block, inversion, supergene are often associated with discrete and complex ecotype, often polymorphic, and could be involved in local adaptation or speciation. The reason for this is that it creates specific pieces of the genome where gene flow is restricted. So it protects co-adapted allele, for example, to make it very simple. maybe I will skip that. It's just that in practical, we try to summarize how we could analyze structural variants in population genomics, like how to optimize the ratio of information data. Validation is needed. There are lots of false positive. And then for the statistics in population genetics, it's important to account for SV specificity or diversity. So a very practical example with the lake whitefish and Atlantic salmon. Here we had assembly or long read. So of course, that's the best data to detect structural variants. You can map an assembly or the other or put long read, but usually you can't get a lot of samples, right? With short read data, it's much easier to sequence many samples and you will capture population level variability. But when you try detecting structural variant, it has a lot of false positives. So one solution we came up with, which maybe is not the best, but it's doable at that time, is to build a graph representing the variants that we're detecting with assembly and long read. On this graph, you would have bubbles that represent inserted sequence, for example. And you can map the short reads on the graph. And based on that, you can call for each variant what is the genotype for the different individual, which help doing population genetics on structural variants. Then what can we do? Well, we can, for example, calculate FST not only on SNP, but also on structural variants. And did we find things interesting with that? Yes and no, maybe. To explain a little bit more of the whitefish system, here we have two allopatry glacial lineages that came into secondary contact in post-glacial lake. And the same moment seems to be a moment of ecological displacement, and one species became dwarf. So now it has a very different ecology from the normal, like feeding on different things, living in different parts of the lake. And there is quite a high reproductive isolation between the two. So if I come back to this, I show you some TE, and actually about 1/3 of these structural variants were transposable elements. And when we looked, I will print everything. Okay. When we look with FST at the most differentiated structural variant, we see that the most differentiated ones were enriched in all the TE family. So what could that mean? Of course, there could be some technical artifacts. We've tried to control for this, but I always have some doubt in the back of my mind. But one biological explanation could be that during the time of allopatry, or because of isolation between the two species, not the same TE have been active, or maybe the same family of TE, but not at the same place. And that's why we see such a structure polymorphism in TE. And it may also relate to what is observed in this species, that when they mate together, the hybrids are not doing so well. And we know that there is a misregulation of the TE, lots of expression of TE in the hybrids and change in the methylation. And methylation usually silences the TE. So it could be that this quite recent event of speciation would be related to transposable elements. Okay. The next study I wanted to present you is about looking at genetic diversity in structural variants and trying to solve this issue that I was mentioning earlier, that the data set are so heterogeneous. So this study is led by Thomas Brasier, my postdoc, and we are trying to make use of public data, such as the genome assembled by Darwin Tree of Life. The idea came that, okay, we can't compare across species because one species is sequenced with short read, the other one with long read. Here, what they do is usually they take one sample per species, a diploid sample, they sequence it with the same methods, going back by a long read, and then they use the same pipeline to assemble a reference assembly. So what we can do is that we can take the reads, map them back on the reference, and ask what is heterozygous. It's a measure of genetic diversity. Of course, it's not as good as you could have for population, but heterozygosity is a measure of diversity. And you will have the same measure in all the species. So we try to detect heterozygous structural variant on SNP. Thomas has made a complex pipeline to look into that, but basically we call SV, we try to check which one are heterozygous, we tried to have a kind of confidence score using, is it detected by 2 tools? Is it well heterozygotes? And then we can custom the filters we want depending on the analysis. So I'm mentioning that because that could be useful for you if at one point you want to detect this. So he ran that on many, many species, maybe, well, many, a few hundreds, and we detect structural variants. And what is showing here, the color, is the density of insertion and deletion, because that's the most common variant, is insertion deletion. And you can see that some plants have very low diversity. So we believe this one may be sulfurs, or maybe the specimen that has been picked is really inbred. But you can see some groups like in insects or butterflies with a very high density of insertion deletion. Yeah, this I already said. Interestingly, for example, we see a very high amount of duplication in the fish, which may be traces of ancient whole genome duplication. We have an interesting pattern with the highest inversion density observed in insects. And I wonder if this could be linked to the way meiosis occur in insects. It's something I would like to dig into, because maybe it's not so bad to be heterozygotes for an inversion in insects, while it could be bad in plants, for example, or in mammals. And the next thing we are doing now with this data set is, for example, testing the Lewontin paradox. So I'm not sure how familiar you are with that. The idea is that the bigger a population, the more genetic diversity you should expect. But if you can see here, that's the expectation of an increase. And when you look at SNP data, it's much lower diversity than what you would expect. So we would like to ask if this is also the case for structural variants. We expect yes, because in our data set, we see structural variant density increasing with SNP density. Just makes sense, more genetic diversity at all variants. But now we want to analyze the subtlety of the different structural variants. And with this big data set, there is much more hypothesis to test. And we are thinking about looking at heterozygosity along the genome, how that correlate with diversification rate, etc. And next step I'm interested in is also compare use population genomic data on structural variants in several species and ask what's in common. Okay. So now I wanted to show you the fly system that you mentioned. So this is the seaweed fly, Celopa frigida. It's living in this kind of decomposing, smelly rag bed that you see. We have them in Brittany, but they also go up to the north. They go up to Svalbard, for example. So they're really resistant to cold. And we've been working on this system for years with Louis and Maren, with my colleague Emma here and Pierre, and now most of my group is working on that. And we became interested in this species because of very big inversion it has in the gene. So what was known from the 70s and the 80s is that this species has a supergene that is affecting size and life history on chromosome 1. And also cytogenetics reported 5 polymorphic inversion, at least in UK population. So when we returned to this system, we were thinking, okay, what can we learn with whole genome data on these flies? And some of the questions, I already showed you that earlier. There are quite a lot of examples linking local adaptation, phenotype, and these big blocks of low recombination. But there are still lots of open questions about what are the ecological and demographic conditions favoring this architecture, what are the functional effects of those blocks, and what's the underlying structure of origin. So I don't have yet the answer to all of that in the flies, but that's some of the goals we want to achieve. So let's start with this supergene, the chromosome 1 inversion. So first we look at this fly in the wild. So it was known, this is data from the 80s in Scandinavia, and they would see that this inversion is polymorphic in all the population, but with subtle variation depending on the habitat. And they also see an excess of heterozygotes everywhere. When I went to Canada, I sampled the population and found the same polymorphism everywhere, and also an excess of heterozygotes in most population, but not all. And when I analyzed the frequency of the invasion with the local environment, I find that it co-varies with how big the rock bed, the habitat is, and how warm it is, and what's the composition in terms of fucus versus laminaria, rock versus kelp. So it seems to be that the very local microhabitat is co-varying with the frequency of the inversion. To dig more into that, we took them in the lab, we raised them for a few generations, and maybe I should have put that slide because you would be interested, but I was comparing, for example, fucus, so kelp and rock, and we didn't see any difference while we expected that. Instead, we saw the same coordinated pattern, which I think was responding to the way we were raising them. We were leaving them a lot of time to develop, which has favored one haplotype, and you will understand which one, the alpha haplotype. But using this generation and we could assess the growth, so egg to adult viability and reproduction success. And basically what we infer is that the alpha allele was not so good for growth. We had a lower survival, but the beta allele was better. And for reproduction, the alpha allele brings a strong advantage. So it takes them, it's quite simple. It's a classic trade-off, right? The alpha means that you take longer to develop, so more risk to die before the habitat is removed and in the lab to suffer from bad conditions. But once they're adults, they're big, they have a good reproductive success. So it's a classic trade-off. And in this case, it leads to some overdominance. And in that case, that's balancing selection emerging from this. There is also an intrinsic advantage of the alpha beta, but this varies depending on the density. So when density is high, only alpha beta, mostly alpha beta will survive. When density is low, more or less all karyotypes have their chances. Yeah, so that's what's called like antagonistic pleiotropy, to have different advantage depending on the life stage. Then we look at these data sets with a lot of genomes sequenced at low depths, about 1,500 flights. The very first thing you can see when you do a principal component analysis is how much this inversion is driving genetic variation. It's restructuring the population. It's no surprise because this region is a very strong linkage disequilibrium. And so we could see where it is in the genome, and it represents 10% of the genome, so it's really huge. It's characterized by very high divergence across arrangement. This is FST between alpha and beta. It's super high. We have also a very high Tajimas corresponding to our excess of heterozygotes. So to put everything together, here we have this inversion that is really two linked haplotypic blocks that are highly divergent, rearranged, that have hundreds of genes. We've looked at expression, and hundreds of genes are also differentially expressed. This has a pleiotropic effect leading to different on life history traits. So we observe this trade-off, survival, reproduction. We observe overdominance. There is also some non-associative mating, apparently, from all the paper, and we could see a difference in the cuticular, hydrocarbon cuticular. You see that's how flies recognize this. And what we modeled and infer is that there is some temporal and spatial variation in habitat quality and availability. And this reinforces the balancing effect. Okay, but this is one inversion, but what else could we find in the genome? So I told you that we can scan the genome to find this non-recombining block. So that's what we did. We found quite a lot of them. Some are still unresolved. I'm hoping to have the new genome to look into that. We found back the inversion we know, and we found another inversion that's quite interesting on the chromosome 4. What we are doing now on the chromosome 1 is try to characterize its structure. From the cytogenetic, it seems to be a nested complex inversion, maybe with different strata. And this is the graph. So now we have several genomes, and my postdoc Jana, she tried to make a graph, and for now it's very complex. So we are trying to solve this at the moment. The other inversion is much nicer, and you will see it's much easier to interpret. So Fantine, my PhD student, she focused on that. She's used first haplotagging linked read. To give you an idea of what this method does is when you extract DNA, you have long fragments, but you need to cut them to sequence. So here you cut, but at the same time you add a tag. So then bioinformatically, you will know that two fragments come from the same area because they have the same tag. But, so that's what is the contact along the diagonal. It's a bit like IC. But here you see another contact, and this is in the heterozygote. And you see that heterogeneous contact that represents the breakpoint of the inversion. It's even nicer with the long read. So what Fantine found is that we have split reads arriving at one breakpoint here that get split and map the other half map at the other breakpoint. And we have split long read that support the two breakpoint of the inversion. So here we have a very simple rearrangement, simply inverted, which is very different to the supergene one that I showed you earlier. And with this, we can also look at it a little bit differently. So I show you the PCA that really represent the block that do not recombine. So here you see the block that do not recombine extend over 5 and 6 to 6 megabase, even a little bit longer. But this is where the breakpoints are. So with this inversion, it seems to be that the recombination suppression extends beyond the breakpoint. And it seems that's something that's also observed in Drosophila, and I'm curious to hear if it's observed in other species. So this inversion, instead of knowing the phenotype and looking at the genome, we found it in the genome, but we had no idea what it does on the phenotype, and we wanted to know. So that was the work of my PhD student, Leah. Like, try to find traits associated with that and understand the link with ecology. So what we knew is that the frequency of this inversion was varying along a latitudinal cline. So it's more, we call it S. So the arrangement S is more frequent in the south, and it's more rare in the north. And we call the other arrangement N, like north. So Leah looked at European population, and she found the same cline, so making really parallel cline in terms of association. Obviously, what varies with latitude is temperature. So we see that the frequency co-varies with temperature on both continents, but it may also be other factors. It's just everything is co-varying with latitude, right? So we went to the lab and tried to do experiments on this. So we had this expectation that fitness would be higher for the S when it's hot and higher for the N when it's cold. So Leah spent a lot of time making homokaryotypic line. So we wanted to get SS line and NN line. And then they did a lot of experiments on that, trying to measure resistance to cold stress. This is with Andrea. They have been making, raise them at different temperature, measure development time, fertility, longevity, viability. So I'm skipping most of the results here, but in short, the only significant effect was on fertility and viability. So it seems that this inversion affects fitness. We would see differences between the two lines, but it was not going in the direction we were thinking. More generally, we found that the S allele seems to have a higher viability, and the N allele had higher fecundity. And the effect was not exactly the same between the two continents. We were looking at America and Europe. So it affects fitness, but the interaction with temperature is limited. And there were differences across continents. So we came back and think, what was our hypothesis? Maybe it's not so right. So I think we were thinking about a simple selection migration balance, thinking that one allele is good when it's cold, the other one when it's hot. Maybe it's that, and we just haven't tested the good environmental factor. Maybe it's daylight, or who knows. But we also think with this effect on viability and fecundity, we could have again some balancing effect. And there is this theoretical prediction that you could also have clines with balancing selection. It's just that one allele is slightly favored and the other one, but somehow both alleles could be present. And we tend to think that it's more likely that we are in this context. So to summarize, we aim to do this link between ecology, traits, and genetics. The association with the latitude gradient is very clear. It's very strong. It's parallel, so it's unlikely that it's stochastic. Seeing this repeated is make it likely that it's selection. But the link with the phenotype is hard. I felt less alone when I look at the Stickelback paper, in which they also try to do experiment, and it was really hard to reproduce in the lab what you do observe in the wild. But they also have very clear association between the habitat and the inversion. So I guess we shouldn't be so surprised, maybe, because the environment is always multidimensional, and in the lab we can only test a limited amount of environments. And when you look at inversion, it's really a complex architecture. There are many genes around. And even here with homokaryotypic line, we did not control the background, so there's lots of noise around that. Yeah, but I would still like to know what traits are exactly affected, I have to say. Okay, so just to summarize, I've been showing you two inversions on which we are trying to elucidate the genetic architecture. For some, we know well the genetic architecture, not the phenotype, and the other way around. We think that both of those inversions are, so we know they are very polymorphic in nature and they covariate with the habitat, so they must be somehow under differential selection and involved in local adaptation. I'm interested to contrast the two, also because I think the balance pattern with very high excess of heterozygotes is a different dynamic than the clinal inversion. So what can it tell us about the evolution of inversion and supergene? And we're also interested to think about how this big old inversion compares to the range of inversion. I mean, we have just picked two, but we have other areas which are weird, possible other blocks, and they are likely more smaller or younger inversion that we don't know. And here is another overview. I hope I've been also showing you that there is this link between the ecology and the rearranged structure. It's not always easy to make the link with the traits, particularly complex traits like fitness. We saw an effect on recombination that expands beyond the breakpoints. And what we are trying to see at the moment is also the impact on how the chromatin is open, or this kind of epigenetic effect, and the link with gene expression. And well, this thing of haploblock I've been showing you, they have been found all over in the literature recently. So maybe there are many more haploblock polymorphism or big inversion polymorphism. But I think we need to be careful also, because sometimes they are not mega base scale rearrangement, they are just low recombining region. I'm not sure what's the time. I have a last part. But... Okay, so I go fast on that. So this is work that was done by Xavier Dallaire, who is a PhD student with Jean-Sébastien Moore. And I've been working with Xavier when I was in Quebec, and he came visiting my lab last year, in which we work on this. And I think he has made two great examples of why it's worth accounting for structural variants in population genomics and biogeographic study. Like, even if you have SNPs, it's relevant to know SV exists. So why accounting for structural variant in population genomics? So perhaps coming back. So Xavier is working on the active chart. That's the beautiful fish here, which is a very important fish for northern community in the Canadian Arctic. And he was doing some basic population genomics and calling SNP. And what he figured out is that lots of heterozygous SNPs were not really SNPs, they were duplications. So that's known, that's also observed in plants. And what you can see here, so if you're familiar, this is a two-dimensional SFS, and you have the spectrum of allelic frequency in one population and the other population. And here you see an excess of shared SNPs, very strong along the diagonal. And most of those are heterozygous SNPs coming from duplication. And when you remove, you filter out this weird SNP that are not true SNPs, you get a pattern that's much more expected. And it was very important to filter them out. And you can see here the FST estimated with all the SNP, and the one when you keep just the canonical SNP, so removing the one that are from a duplication and that this mismapping in the genome. And the estimation of FST are not at all the same. So it's important to know when we quantify genetic variation, genetic differentiation, what are the variants we take, and whether we have some spurious variant like this. The second thing that Xavier did with this data set is look at the biogeography. He was interested in the recent demography of this species. So what they knew is that this is from the mitochondrial genome. So they knew there was an Atlantic lineage and an Arctic lineage. And the Atlantic lineage was supposedly only in Greenland and the easternmost part. But when you look in the genome, you could see some patterns of introgression. It's here. So this is the whole genome ancestry between the Arctic and Atlantic. And you see that there is more hybrid zone that's going much further east. So all of this hybrid zone and introgression is completely reshaping the distribution of genetic diversity. We were interested in haploblock, thinking there could be inversion, could be rearrangement. So we did the same analysis, scanning the genome with local PCA, like I showed you. And Xavier found a lot of this area which are putative haploblock. And then when you look at those windows, you would see the same pattern. So they will have one haplotype, that's the blue one, that is more on the eastern part, and the yellow haplotype, which is the same as the European one, that is more in the east. So we started thinking there can't be so many rearrangements showing the same. So we rather think here that because it's recent secondary contact, it's just that recombination has not been important enough to reshuffle this. So any region where recombination is a bit lower would keep this long haplotype. In this paper on the bird, they have another representation of that. It's simply that yes, haplotype are longer when recombination is low, and we keep this haplotype coming from two different ancestry. So sometimes it's argued there could be barriers to reproduction. That's why they are maintained like that. Sometimes it could just be neutral. We just need more time. What we decided to do in this species, because we couldn't know what they are, the genome is not good enough to look at rearrangement, but Xavier thought maybe they have information about the species, about the biogeography of the species. So he was trying to infer the colonization. And when you run this kind of program that try inferring past demography and recolonization of biogeography, you would find that everything was coming from the south. But this program assumed no introgression. So we were not in the good condition for that. So he thought maybe I could focus on the haploblock where I have almost pure northern haplotypes. And when he looks in the haploblock, we found a different insight, which maybe suggests something coming from the west. So this would need to be confirmed, but I guess the message is this haploblock could bear some signal, some information about ancestral population that is lost by the recombination elsewhere. So concluding now, so I hope I've been showing you some of the properties that make structural variants an important player of evolution, because they cover a large part of the genome, they can be big and have a strong impact on fitness, they can evolve fast, and they can impact recombination. Here we've discussed some of the methods to detect genotype and analyze structural variants, and a few examples of small and big structural variants involved in speciation, local adaptation, and the balanced polymorphism. And I hope you keep in mind some warnings about comparable data matters for structural variants, that as soon as you find a haploblock, it's not necessarily an inversion, and even in SNP dataset, we should be careful because of structural variants. And I think with this, we are opening a Pandora box looking at all these variants. Will we find the same information as regular mutation or different thing? I don't know yet, but we know that some structural variants bear extra information, and now we can do large-scale comparison and we can address many open questions. I've listed some of the ones that I'm interested in at the moment, but thinking what are the characteristics, the property of structural variants that could make them matter for evolution. I think I will stop here. First, I'd like to thank everyone that was involved here and there working on the project, and thank you for your attention.